Thanks. All right, so this morning I'm excited to introduce Dr. Sally Otto. Dr. Otto is a Canadian research chair in theoretical and experimental evolution and a Killam professor at the University of British Columbia in the Department of Zoology. Her research focuses on using mathematical and statistical modeling to understand how evolutionary processes give rise to biodiversity. And she's an elected member of the National Academy of Science and has received a number of the top awards in her field, including MacArthur and Guggenheim Fellowships, the Darwin Wallace Medal from the Linnean Society of London, and the Sewell Wright Award from the American Society of Naturalists. On a personal note, I'm really excited to have her here because I found a lot of solace during grad school in her book, Biologist's Guide to Mathematical Modeling in Ecology and Evolution, which I found to be extremely useful for introducing modeling. And I still kind of lug it around to consult today. Dr. Otto is part of the British Columbia COVID-19 modeling group and has published a number of important works related to the evolutionary epidemiology of SARS-CoV-2. So please join me in welcoming her this morning.
Thank you, Spencer. It's a real delight to talk with you. And I know you've had another team member, Caroline Colleen, speak with you in the past. And so what I'm going to do is kind of give you a potpourri of various thoughts on COVID, and then we can choose our own adventure and kind of go into more depth in any of these aspects. So I'm just going to share my screen and start launching in. Let's see, let's go here. Okay. And how do I get to full screen? There we go. So I'm going to do this in two parts and talk a little bit about the BC COVID-19 modeling group, which I think is quite similar, perhaps, to the team that you are all in. And then I'm going to move into evolution of SARS-CoV-2. So in BC, probably as in many jurisdictions in Canada and in the United States, there was very little modeling capacity within the government. And that led the BC CDC to reach out to colleagues at both UBC and SFU. So we started a team back in March of 2020 that would basically provide modeling support for the question, at that time, the questions coming from BC CDC. And I mentioned this background because at first we had questions kind of aiming at us all of the time. And there was a lot of modeling information going back and forth. At some point, that kind of two-way street turned around a little bit, and there was less interest in what the modeling projections were. But the fact that we were an independent body that was able to continue to talk and respond as a pandemic evolved, I think has been really important because we weren't kind of embedded within BC CDC, we were independent, and that allowed our team to continue to function and act, even when the news wasn't always good news. So we've been meeting we're now down to once a week, but we've been meeting twice a week pretty much for the whole pandemic, similar to what you all just were doing, reviewing relevant data and articles, having expert presentations, presenting new models for feedback with each other, and providing advice. Slack has been enormously helpful, and both in this communications and community building. We've had 12 public reports so far. And just scan over this, but summarizing articles, having experts come. And one of the nice aspects about this team, we don't work by consensus. Some of the other modeling teams work by consensus, and they have to agree. What we tend to do instead is approach our models in different ways. From compartment models to differential equation models to various kinds of statistical fits. And I think from that, by presenting all of those analyses in our reports, then you can see the uncertainty emerge and the kind of range of possibilities emerge by comparing models. So we don't attempt to have just show one prediction or one outcome. And what we've been doing then is providing projections for the public and for public health, addressing policy related questions as they come to us, expanding theory to better fit SARS-CoV-2, as I think many in this Zoom call have done as well, and filling in gaps about the biology, for example, serial intervals or estimating selection coefficients. So inside of our team, Caroline came and talked to your group. Many of her team members are also in the BC COVID modeling team, and they use one of these compartment models that account for both individuals able to work from home and to be have lower contact rates, and for others that are essential workers and or for other reasons don't reduce their contact rates. So this allows, incorporates heterogeneity in people's behavior. So we've done this is where the potpourri starts. It's really a bunch of different questions that have come up over time and that have been addressed by different models. One that was influential in Canada was talking about there was a real focus on whole genome sequencing and subsampling of types. But when alpha appeared in Britain, we pointed out that if you that there's a real value for fast PCR sampling of a higher fraction of cases in order to detect variants of concern while they're at low frequency. So that led to a kind of rapid assay PCR tests in BC and other provinces. We've looked at contact tracing. Contact tracing is fantastic, but it is got, we were interested in just how to reduce capital R. And it has some limitations, and that's what we were focused on. One of their big limitations, given the delay until symptoms before you know your index case, and then delays in stopping and the delays in the contact tracing itself, preventing secondary cases from transmitting. So anyway, this analyzed just given how many people are in the contact tracing program, the percentage of people in the program, and those delays, just how much could contact tracing on its own reduce capital R. And you can kind of see it can reduce capital R values that are near 1 to below 1, but it really struggles on its own to reduce very high R rates. And another thing that we've been doing recently, but more with the engineering members of the team, is switching from pandemic predictions to more of a feedback control perspective, where there's so much uncertainty in any pandemic like this one. And so prediction is always going to be struggling with that uncertainty. If instead what you do is say, okay, there's going to be uncertainty, but we're going to use some measure of cases or hospitalizations. And as it rises, then we use the cumulative evidence of how far off we are from a particular target to ramp up levels of control. And this is like a thermostat where, it won't necessarily go on and off just because somebody walks by the thermostat, but as the evidence accumulates that the temperature is higher than or lower than the target, then eventually more and more of your heater or your air conditioner will start working. And it's interesting because that even if there's uncertainty in transmission rates, in who's the age structure, the stage structure, the uncertainty in vaccination efficacy, all of those kinds of uncertainty. If you take this ramping up approach and with feedback, then we show that you can reduce the overall number of cases expected in simulated pandemics, but also reduce the total amount of restrictions. Because what can happen, it did happen here last summer, you can have very, very few cases and really high restrictions for a long period of time. And that also is a mismatch to what is needed to control. Anyway, I think for those, some of you might be interested in these ideas of switching from pandemic prediction to pandemic control measures and using theory about feedback control, like how do you build a thermostat so it works better, that kind of thing, to also help us understand how to control pandemics. So I mentioned reports, and our reports are, these are snapshots of what are happening. So this is just a couple of slides pulled from our recent report. And I just put this one in, Spencer, because it relates to that issue of how vaccination coverage in different communities, these are different health, this kind of smallest level of health authorities in British Columbia, and just how much protection we see in those regions that have very high, this is of the eligible 12 plus adults, what fraction are vaccinated? And we, again and again, over the last few months, we see much lower levels of cases per 100,000 in the most vaccinated communities. These numbers go, different communities are riding a roller coaster, so the actual numbers in different communities are going up and down. But again and again, it's about 3 to 5 times more likely to, we'll see 3 to 5 times more cases in those least vaccinated communities. One other thing that we're highlighting in our last report was just how important children under 12 are likely to be right now in British Columbia in controlling this pandemic. So of those people that are this is an age distribution of who is unvaccinated in BC. And you can see that now it's about 50% of the unvaccinated population in BC are children under 12. I mean, we do have a really remarkable vaccination campaign. We're almost up to 90% of the eligible population of the province, at least first dose vaccinated. So this is pretty extreme, I think, but it's great news. But you combine that with a contact, this is a contact matrix between the different ages, just pictorially representing the fact that children tend to interact with other children. And you put those two together, and this do a sensitivity analysis. What this does is it says, if you're going to change measures impacting any one age, which ones are going to matter most to the growth of the pandemic, which are going to affect R the most. And you can just see like it's 20 times higher sensitivity to whatever we do in the under 12s. And this accounts for the fact papers showing that youth are more likely asymptomatic and that there's less transmission from asymptomatic cases. But this is also a very different picture from last year. This is what we had last year before the vaccination campaign. And it led to this narrative in BC that the measures in schools didn't really matter much. And that was last year's narrative. So we're trying to do these type of analyses to really help public health officials recognize we are in a very, very different situation now. And schools will be really important drivers in the pandemic in British Columbia with such high vaccination rates. So this is just an example of how this is the little r, the daily growth rate, as a function of changes in contact rates. So this is continuing with current contact rates, and this is if you increase or decrease contact rates. And just how it's just another way of saying it really matters what you do with the school age kids right now. So that is a little bit about what we've been doing kind of as a modeling team, and just some snapshots of that. I wanted to talk about some of the work that colleagues and I have done on the evolution of SARS-CoV-2. As you all know, within weeks of the initial reports from Wuhan of a new respiratory illness, scientists around the world were already searching for signatures of adaptation to humans within the genomes of SARS-CoV-2 and trying to figure out how selection was acting and how this virus would evolve. And this is a kind of a summary, and we can decide how much time we want to spend on the modeling. But modeling work that I did with Troy Day and various other colleagues in a couple of papers in Current Biology really emphasized that early on, when there was very little immunity within a population, most individuals are susceptible. The selection was primarily for increased transmission. And one other prediction which we made was that selection was predicted to slow when control measures reduce transmission rates. So those variants that have a transmission advantage, you basically take away their advantage if you're in lockdown. And so we've subsequently seen evidence of that the selection coefficients very, very much dependent on those control measures. And in times of lockdown, the selection coefficients have gone down for the variants of concern. Now, however, selection is increasingly, now we have an increasingly high fraction of the population that's immune, largely through vaccination in Canada. And selection then switches and favors variants that are able to infect at a higher rate those individuals that are immunized. And so we can go into some more details about those kind of models. One thing that is important to emphasize in both of these all of these models, is that for SARS-CoV-2, there's very little direct selection on severity, on virulence, on whether or not this virus kills us or not. And that's because those severe symptoms manifest after infectiousness, after the viral load has come and gone. And so that means that whether or not this virus kills us is pretty much irrelevant to the evolution of this virus. A lot of those severe impacts are caused by immune reaction sequelae to the infection itself. Now, as you all know, we do see differences in severity, but those are always pleiotropic effects, we argue, for what selection was actually acting on, which was increased transmission rates. One consequence of this, though, is that we don't predict that it will always be the case that variants of concern will necessarily be more severe. There can be selection, the pleiotropy could work in either direction. And in particular, for example, this is the model that we use: susceptible, exposed, but not yet infectious, pre-symptomatic, infectious, asymptomatic, and recovered. And in this model, if most of the infections are happening in the most of the people circulating are asymptomatic individuals, and that the individuals that know they have symptoms are isolating, then it could be selection for higher asymptomatic infections of lower severity. So it could go either way, and we'll have to pay attention. We don't have a crystal ball to say whether or not severity will increase or decrease with every variant. And there's good reasons to think it'll go up and good reasons to think it will go down. So this is where I'll come back to modeling if you want to be able to understand more how selection acts on all of these components of the basically life history of the virus infections. But the main points were that evolution favors higher transmission and slower progression to symptoms, basically to keep these pre-symptomatic individuals out and about before they know that they have symptoms and self-isolate. Virulence and recovery rates of infected class are nearly neutral, as I mentioned, because that happens late in the infection. And so evolution of virulence for SARS-CoV-2 is more likely to be a byproduct of direct selection on mutations affecting transmission or progression. And just in case, that's, the classic story is that virulence is selected to go down. And that's the case when virulence and is kind of those severe side effects are earlier acting. And so if the severity really limits transmission, but that's not really applicable susceptible to SARS-CoV-2. And of course, we know also of diseases that are flipped, where transmission happens through death and disease, and through the the virus is really transmitted through only after people have those severe diseases, severe symptoms. And Ebola is a little bit more like that, in that a lot of transmission happened during death burial ceremonies, and because of that there was selection for higher severity. Anyway, it depends on the disease. There's no across the board reduced severity is always how a virus will evolve. That is commonly thought to be true. It's commonly said to be true, but it's really important to emphasize that it's not true, period. And for SARS-CoV-2, severity is almost under no selection itself. We've turned more recently to looking at how more and more resistant variants are likely to arise, and the selection that they'll be under. So here's a really simple model to think about that. If you have a normal SIR model with infected individuals and mutants, and really you need to think about this as a series of I1, I2, I3, I4, where every single mutation that arises is just a little bit more likely to infect an individual that was previously resistant, either resistant through vaccination or resistant through infection. And so we know that escape is not one single change that has to happen. Our immune responses are extremely robust and varied, and that means that we've got lots of antibodies and B cell and T cells that will react. There's not going to be a one-step mutation, but nevertheless, selection will favor the increasing diversification at sites recognized by our immune system. So what's the first stage in this? And there's the evolution of these increasingly escapable variants depends on two things. One is the first appearance. If you don't have any mutations that can infect a resistant individual, then there's the waiting time until those mutations arise. And the mean of that in a community with a fairly constant level of infections is this. And then you have to ask, well, how does the partial escape mutant spread relative to a regular virus? And that, of course, the main selective advantage it gets is these infections of resistant individuals. So it has a different, an additional component. Not only is it able to infect susceptible individuals, but it's able to infect at a partial rate, this P, previously resistant individuals. So that means there's two things. If you want to slow or prevent the evolution of resistance, there's two things you have to think about: preventing mutations from appearing, and then the second stage of preventing spread. Mutations. So what can we do to reduce mutation rates? Well, that's hard, but one of the things.
Quick question.
Yeah.
Sally, just on the previous slide.
Yeah.
It's a really cool model, but I guess I'm thinking, like, as long as you have rho or P, I guess, beta RI greater than beta IS, I guess maybe some combination essentially of I star bringing out of R and bringing out of S, you're going to see dominance, right? Are there any like interesting properties of dominance that this model is explaining, or is it all about time to dominance?
No, so you raise a very good point. Basically, the R of the escape mutants, if there's no side effects of this, it just has a selective advantage by this amount, because it's increasing the number of viral offspring per viral parent by the amount that it can transmit to resistant individuals. But these mutations could have, if it's a mutation in the RBD, it could actually be less able to bind to cells, and so there could be trade-offs which this does not capture. I mean, if we knew about the trade-offs, we could measure it from this model, but it could transmit less, for example, in susceptible individuals. So there could be a lower, the beta could be lower. And then it's a question of, is the advantage of infecting resistant individuals high enough relative to those costs? Yeah, so
Yeah,
that's
interesting.
a really important empirical question. Mainly the model helps you organize and kind of calculate what those
Right.
costs and benefits would be.
Cool. Thank you.
Yeah, and so one of the things I wanted to highlight that came up, this Merck drug is one that is one concern about mutation rates. So we need to do whatever we can to stop especially multi-step mutations from appearing within populations, doing everything we can to support immunosuppressed patients if they by vaccinating everybody around them as much as possible, but then also monitoring them if they do get COVID. So, but molnupiravir, I don't even know how to pronounce it, but the new Merck drug is a nucleoside analog, which is meant to lead to the air catastrophe, which there's so many mutations are introduced that the virus fails to replicate. And that's a really great idea for a drug. But if there are ever tissues or patients in which the dose is not quite high enough to lead to the actual catastrophe, it could just cause lots of errors. And so I am quite concerned and think that this is an important policy point, that the drug should be used and with individuals fully isolated and closely monitored for any onward transmissions, particularly onward transmissions of highly mutated strains.
Do you know if what the data are from the clinical trials on the emergence of at least molnupiravir resistant viruses after following treatment?
No, I don't. I don't think that I was told that they don't have that type of data with the Merck studies, that they are measuring efficacy on keeping people from dying and not looking at onward transmissions. But this is what I recently told the public health officials in Canada, is that we need to elevate this as a request for the company and a request for public health officials. There are other drugs that act as nucleoside analogs, but almost all of them are chain terminating, meaning if it gets incorporated, then the polymerase just falls off and dies, and it stops the prevention of RNA replication. So molnupiravir instead gets incorporated, it gets mismatched and causes cytosine to, I can't remember which one, I think it's like half AU mutations. Anyway, but it gets incorporated. The error correcting machinery of SARS-CoV-2 doesn't recognize it as a problem. And so the whole cycle of replication can continue. So anyway, I do think that there is, this is wonderful life-saving drug. I think the mortality rate was halved. I'm not saying don't use it. I'm saying use it under really, really careful monitoring, because the last thing we need are more doses of mutations leading to just this virus being able to explore genotypic space much more quickly. Of course, everything else that we do to reduce case numbers also reduces the appearance of new mutations. And then the second phase, how do you spread it once it appears? Then you basically are trying to reduce the fire and reduce the fuel. Reduce the fire, and that is trying to identify these individuals that are able to infect immunized individuals and isolate them as best you can, reduce by detecting and isolating. And that requires, us, one of the things that we need, and we're really struggling in Canada to get, is more information about genomes and vaccination status. Nobody has that information across Canada. And so we can't really say, oh, we've got to watch out for these variants because these are the ones that seem to be heritably infecting vaccinated individuals. And if we can identify that signal, that would help us isolate variant cases more. But the other thing that can reduce the selective advantage is you basically want to reduce contact between infectious individuals and immunized individuals. So strategies like in that town that you were just talking about, where they did this pulse of vaccination, is a more efficient way of getting of reducing contacts between susceptible and infected individuals and resistant individuals. This kind of slow and steady increase in vaccination levels is basically the best scenario for escape mutations to spread, because it just maximizes the amount of time in which there is this selective advantage of getting into resistant individuals. I think that is enough. And then I will just talk very briefly about two other kinds of things that we're doing. Like you all are scanning genomes for signs of new variants. I will just skip that. So we're looking within Canada for Delta subvariants that are arising. There's some signs of selection within the subvariants. So we're just monitoring and tracing that. I'm not allowed to tell you, for some reason, I'm not allowed to share with you which actual subvariant strains these are. Hopefully that approval, so there's a huge amount of privacy restrictions that's driving me crazy in Canada. And these are public health authorities basically protecting the privacy of this bloody virus, and it's annoying. And the other, but hopefully we'll get that all sorted out. Yeah.
Sorry, can you repeat what this is showing us, this graph?
So this is showing over this summer, the spread within Canada of different subvariants of Delta. And so you expect an S-shaped graph. Sometimes you don't see that. Like this one's not very S-shaped, but that's because it is S-shaped in some provinces and it's not present really in other provinces. And so I can't show you that the province by province S's, but we're seeing repeated spread of some subvariants across provinces in the kind of S-shaped curve you'd expect for a selectively favored variant. The selection coefficients, I should say, are about half of that alpha had over the wild type. So we're not talking game changing huge amounts of increased transmission, but we don't want any increase in transmission. We don't know enough about the causative mutations in these subvariants either. Delta is now fairly diverse, and so each of these subvariants carries half a dozen or a dozen differences, making it hard to know exactly what's important. The other thing we're looking for are recombinants. And I think other people may be doing this in a wiser way. All I'm doing for this is, there was a NSP6 deletion that was found in alpha, beta, gamma, and lambda, and it wasn't initially in Delta. So I was concerned that it was a beneficial deletion since it was found in all of the other variants of concern. So we've been scanning for, are there any variants that carry Delta spike and that particular deletion? So this is cumulative sequences in GISAID over time. This is cumulative, which means it's not that bad. It looks like it's growing a lot, but that's just because there's just a steady increase in number of sequences like this. So we're just keeping an eye out for whether or not any of these variants are going anywhere. We are seeing community spread. So that Delta with the NSP6 mutant will appear in a community, and then we'll see it in other places in that community for subsequent weeks, particularly in Italy and Turkey. I don't think it's alarm raising yet because we're not seeing it spread really fast. But again, this is hard to know if, I said it was spreading in the community in Italy, and then it seems to flatten off. Is that because we just don't have genomic sequences from Italy from August? Or is it because it actually doesn't have a selective advantage over the other types? I'm going to stop there. Sorry for being all scattershot, but it's like, I want to share everything with you all. nothing in enough depth to do anything with, but maybe that will, I can at least give you pointers to papers, or we can take questions now.